name is Jamiel, and I'm a microbiology student at the University of Massachusetts Amherst, currently researching in Dr. Michelle Klingbeil's lab.
Our lab studies the parasite known as Trypanosoma brucei. T. brucei causes serious vector-borne diseases in vertebrate animals known as trypanosomiasis. This disease can be fatal to humans, cattle, and other vertebrate animals if left untreated. Research suggests that one of the major factors restricting economic development in Africa is the negative effects T. brucei has on agricultural, veterinary, and forestry development. Trypanosomiasis is causing livestock in sub-Saharan Africa to become sick at a rate which costs the continent up to $2 billion a year in revenue. Current treatments for cattle are difficult to administer, expensive, and extremely toxic. Human treatments can also have severe toxicity and side effects in human patients. Because of this, trypanosome researchers are actively looking for a safer and more effective drug treatment for trypanosomiasis. T. brucei contains a complicated network of mitochondrial DNA known as kDNA. This network contains two types of circular DNA structures, minicircles and maxicircles. The replication of this network is performed by intricate mechanisms completely unique to trypanosomes. The organisms release the minicircles into an area outside of the network. There, the minicircles are replicated, transported, and reattached to the network at a different location. In order for this process to be successful, numerous replication proteins must work together in synchronicity. The survival of T. brucei requires the functionality of this kDNA network, and therefore the functionality of all of these proteins. By designing a way to effectively disrupt mechanisms within kDNA replication, a new cure for trypanosomiasis may be established. However, the process of kDNA replication is substantially different from any other form seen in other organisms. Because of this, much more needs to be learned about the process by which T. brucei replicates its kDNA. My research aims to determine whether the specific proteins thought to act together during kDNA replication are coming in direct contact with one another. These interactions can be discovered using a yeast-2 hybrid system. This system mates two yeast strains together on selective medias to examine protein interactions. A protein of interest, known as the bait protein, is fused to the DNA binding domain in a strain of yeast. The second protein of interest, known as the prey, is fused to the activation domain in a second strain of yeast. If the bait and prey proteins interact when the yeast is mated together, transcription and activation of specific reporter genes will occur. 3 of the 4 reporter genes make it possible for the yeast to grow on media that would otherwise not support growth. The final reporter gene encodes for an enzyme that leads yeast colonies to turn blue on specific media. The growth and color of the yeast colonies on the selective medias show the reporter genes have been activated, meaning that the two proteins have interacted. My research will use specific kDNA replication proteins in T. brucei to determine if these sets of proteins are interacting during kDNA replication. By determining whether or not these specific proteins are interacting during kDNA replication, we'll have a better understanding of the processes that are occurring. By better understanding these processes, we can create a drug treatment which will disrupt these processes and cause the kDNA network to fall apart. Establishing a better drug treatment will not only increase survival for infected humans and cattle, but will also help to alleviate the strains that T. brucei puts on Africa's economic development.